The important synopsis of Chapter 3 is a great introduction to the history of research on tumor viruses, which changed our perspective on how cancer operates inside our cells. Prior to the discovery of tumor viruses and their mechanisms, we were at a loss as to how cancer develops. There were many ideas. We didn't have the tools to investigate because it seemed so sophisticated in the 1900s. But tumor viruses have opened our eyes properly since the 1950s. So keep that in mind. And the second component of Chapter 3 is to illustrate to us how viruses, tumor viruses, operate within our cells. They fall into three basic classes. Let's get started with a general overview of some of the learning outcomes. So you can see, in order to match the examinations, you have to do a lot of construction of words, like describing and explaining, and then distinguishing between various components of tumor virus biology and history. So make sure you keep that in mind as we go forward. Every living thing, from bacteria through fungi, through plants, up until you get to animals, including humans, have viruses which infect their cells. As the slide says, humans have so far discovered over 500 different types of viruses which infect us. Some of them are listed on the left-hand side of this slide. The important thing for you is to be able to try to discriminate between diseases caused by viruses, as listed here, and those that are caused by non-viral agents like bacteria, protozoa, and other types of parasitic agents coming from the animal kingdom. Remember that viruses themselves are considered by many to be non-living, because if you apply Mr. Gray Plus, something that we learned in Bio 2, you will discover that it does not, or they do not satisfy all the criteria, all 7 criteria for Mr. Gray Plus. The biggest failure is at the level which we call cells. So viruses are not made of cells, and therefore they cannot be living. Other biologists are beginning to wonder where the gray line lies between life and non-life. And things may change in the future, right? As we discover new types of viruses, which are in that gray area between what is a cell and what is not a cell, believe it or not. But let's see what happens in the future, right? As far as we're concerned, viruses are not living. So what do we need to know in general about viruses? Well, that's on the next slide. So viruses come in different sizes and shapes. Some viruses are almost as big as the smallest bacteria. But most viruses are in between the smallest viruses, which are no bigger than molecules, and most of the viruses are in between those two sizes. Their shapes can vary depending on their biology and their evolution. Some viruses, like the poliovirus that you see here, have a standard spherical shape. That's their capsid, right? The capsid is basically the container which contains their internal nucleic acid and possibly some internal proteins. Other viruses, like these T4, which infect E. coli, they have complicated shapes with heads and tails. And yet other viruses, like these tiny, tiny, tiny adenoviruses, they are hard to visualize, but they are surrounded by an odd architecture of proteins that enable their life cycle to continue. Most viruses have simple life cycles. Their job is to latch onto a cell, a target cell, and that's determined by the compatibility between the surface of the virus and the surface of the cell. So when you get a docking between the two, then the virus springs into action by some type of spring-loaded mechanism, and the virus ends up either being internalized through phagocytosis or just injecting its nucleic acid into the cytoplasm of the host cell. There, the machinery of the cell is sequestered for the benefit of the virus. So basically, the virus takes over the entire cell to make progeny viruses. And that involves, in this particular case, we have a double-stranded DNA virus. And in the ensuing chemical changes, not only is DNA made in bulk to match the chromosome of the virus, but proteins are also made through the ribosomal system of the cell. And then assembly takes place automatically of the progeny virus particles, which are then somehow eliminated from the cell to continue the life cycle of the virus. So out here and down here, the viruses will be considered non-living. Inside, that then becomes an arbitrary designation. There's no standard picture we can show you, but here's one that talks about this type of virus. So you can see deep inside we have the nucleic acid. Then we may have some proteins that are useful for supplementing something that the cell can't supply, in this case, an enzyme called reverse transcriptase. Then there's a core made up of proteins that protects the nucleic acid. And there may be a shell that's also made of a combination of sugars, lipids, and proteins, which gives the virus its outer coat. And then these are able to have the necessary components in order to attach and then sequester cells. Some of the smallest viruses we know, they only carry a handful of genes, sometimes as few as 5 genes. And those genes are enough to fool the cell into becoming a system of viral progeny production. Okay, so it's amazing what viruses are. In a simple sense, viruses are cellular parasites. They have their own reason for existing, and they do so quite successfully. What we want to learn in this particular chapter is, do some viruses cause cancer, or at least the formation of tumors? And that is the main gist of the stories that this chapter tells, right? And it's true. Some viruses, not all, are able to induce tumor formation in their host cells. As a designation of what a tumor is, it's a cell that grows out of control by itself, multiplying through its cell cycle without any restraints, right? Uncontrolled cell growth. When the cell multiplies, then the number of host cells for the virus multiplies. So this benefits the virus too. The textbook focuses basically on the very first virus that alerted some biologists to the life cycle and the behavior of these tumor-causing viruses. So Rous sarcoma virus, which is a virus of birds, which causes a sarcoma in the breast tissue, was found to be transmitted through a very, very fine filtrate. The filter was powerful enough to prevent the passage of all living things. So whatever was passing through would be something smaller than a living cell. And that's indicated in this case. So once the filtrate was collected, that was made from a pre-existing tumor, then that could induce tumors in subsequent subjects that were injected with the filtrate. So that's the beginning. That's the alert that took place. And Rous was one of the scientists who got credit for discovering that. So his conclusions were that the particle was so small that it was smaller than known bacteria. The particle was capable of rapidly multiplying within the host. And the word rapidly is going to be important because, as we'll see later on in this lecture, some viruses cause tumors rapidly, and others, they cause tumors more slowly. And yet other viruses don't cause tumors at all. And others rarely, right? And the third thing is that these tumors could be induced on a predictable timetable. That means that the researchers knew that the transmitting particles were responsible for causing the tumors. That correlation was very important. The next thing to understand is that in the case of RSV, the first indication that it was affecting the morphology of cells was the way that cells were seen to be growing under the microscope. So cells, when they become transformed by this particular type of virus, they change their phenotype and they become more rounded and they pile up on top of each other. And they have a few more characteristic changes that we'll look at on the next few slides. Regardless, the normal cells that are not yet infected by the virus and transformed, they grow in a typical phenotypic fashion that's characteristic of non-transformed cells. So these cells are transformed and these ones are not. Whatever the virus was doing, it resulted in not just one phenotypic change, but a number of phenotypic changes that are all characteristic of the tumorigenesis process. The next observation is that they become contact independent. So most cells in your body, once they make contact with an adjacent cell, they stop dividing. It's just a mechanism to stop the cells from growing out of control. So once they touch their neighbors, there's a signal transduction process which prevents the cell cycle from continuing. Cancer cells, tumor cells, don't do that. They will continue to grow even if they're not making contact with their neighbors or even when they are. So they become contact independent, okay? And that's a characteristic sign of a tumor. In addition, there's something called anchorage independence. Most cells, when they grow on a plastic dish, they need to be in contact at some point in the surface with the substrate. Cancer cells or tumor cells are able to somehow overcome that requirement. So they become anchorage independent. That means they can grow now in suspension and in multiple layers away from the surface of the substrate. Normal cells require some growth factors in order to induce cell division. Tumor cells somehow switch those mechanisms off or alter them in some fashion. So there's a reduced or completely absent requirement for these growth factors. Tumor cells can also reach high saturation density whenever they grow. So the number of cells per unit area is greatly increased, probably because the tumor cells don't get to the same size as regular cells. And of course, the spherical nature helps increase the stacking of these cells. Number 6, they have an inability to halt proliferation in response to the lack of growth factors, right? So most normal cells, if you take the growth factor away, they will stop dividing. These particular transformed cells, they may reduce the size or the level of cell division, but they won't completely stop. So they continue to either generate their own growth factors or they become growth factor independent. Cancer cells are growing at such a high rate because their primary purpose is to divide, and dividing cells need a lot of fuel. So one thing that human cancer cells do is they have an increased need for glucose. And this is indeed one way that you can do a PET scan to detect the presence of tumors inside a patient. But we'll come back to that in a different chapter, right? And the last one, the ability of tissue to induce in another successful or compatible host, the ability to grow is a feature of these types of tumors, right? So if you take some tissue from this mouse and you transfer it into a similar genetically bred mouse, you can get that second mouse to develop the same types of tumors. Normally that wouldn't work because of the immune system. So you can see, so far we have 8 different changes that are the result of the infection of cells by these tumor viruses. Pretty powerful stuff. So in the next few slides, we just demonstrate from the textbook some of these features, right? So there's a nice video that goes with this module, so please watch that. That focuses on this particular parameter, and it explains how normal cells and transformed cells differ in this way, right? Very important. And the most important word is the word focus, right? So we have a focus developing right there. So that's a focus, and outside that region, we don't have a focus. So focus is simply a stacking of cancer cells, and sometimes the foci can become so large that you can view them with the naked eye. One of the most ingenious things that was tested by the scientists in the past was the temperature sensitive nature of different types of host cells. So what you can see here, we have these cells, and they are infected with Rous sarcoma virus, and at one temperature they are transformed, and at a different temperature they go back to normal phenotype. And then you drop the temperature back down, and then the cells become transformed again. So whatever is happening inside these cells must be an ability of the virus, because the virus is the one that's been made into a temperature sensitive entity. Okay, so the permissive temperature is 37 degrees and the restrictive temperature is 41 degrees. So this ability to shift up and down and get the cells to respond tells you that something about the genetics of the virus, or it's at least its products, the proteins that it carries, must be responsible. And that was an important indicator to answer some questions about the etiology of viral tumors. So this experiment is talked about in quite a lot of detail in the textbook, and it's highly recommended that you at least interact with that section in case in the future there's a test on that material. So these are the concepts that we've covered so far, right? And we'll continue to cover. So there's 2 types of viruses. There's actually 4. There's single strand and double strand RNA, single strand and double strand DNA. The question is, which ones of those are able to cause tumors, and which ones are just simply lytic infections? Lytic, the word lytic simply means the virus goes into the cell, takes over the machinery, produces offspring without causing any tumor formation. The word retrovirus is applied to those viruses which can cause the transformation of cells through a mechanism that we'll talk about very shortly, right? And the result of a retrovirus being tied up with the genetic material of the host cell, we call that a provirus in that configuration. So that's very important. We'll also talk about oncogenes. Oncogenes are genes that are defective in some way, and they lead to the formation of tumors. But the normal version of the gene, called the Proto-Oncogene, that exists, does not cause tumors under normal circumstances, right? So it seems that normal genes can become mutated to become oncogenes. Maybe that's the case. We'll talk about Sark and Mick as 2 examples that you need to learn. And these 2 are talked about again and again in the future. And the most important thing from this chapter, other than the biology of viruses, is going to be a mechanism that viruses use of a certain type called insertional mutagenesis. So the word should be pretty obvious to you now. Mutational means they cause mutations in the DNA of the host cell. Insertion means that something the virus is doing must be inserting itself into the chromosomes of the host cell. And that's exactly what we'll talk about later, right? And then we'll classify viruses into different types of tumor viruses. Maybe the quick ones versus the slow ones. Maybe the retroviral ones versus other ones, right? And then finally, we'll have time to talk about the latest advances in viral biology and their use in trying to perform gene therapy because of their life cycle. So there's a few things to get through, and let's go through a little bit faster. So SV40 is a vertebrate mammalian virus that was discovered in monkey kidney cells, right? And it was used, it was discovered as a contaminant, actually, of the polio program, the immunization program to prevent the polio virus from spreading amongst the human population. And as the article says in the textbook, later on they monitored these patients, and it didn't seem to cause any undue number of tumor formations, which was good news. But the important thing is the virus particles can be transmitted amongst a number of different hosts because their cell surfaces are compatible, right? So SV40 is a zoobiotic agent. That means it can transmit itself across many species. SV40 is not lytic, right? So in non-permissive cells, so this is important now. So as we said to you earlier, the virus and the host cell have to dock together. And if the virus is evolved to dock onto these cells are known as its permissive cells, the normal cells, right? But sometimes the virus can also enter somehow non-permissive cells. And those are going to perform, or the outcome is going to be different in that case, right? So permissive versus non-permissive. So remember that. So this is referring to non-permissive cells on this side. So when the virus enters non-permissive cells, it doesn't cause the death of the cell because those are non-permissive cells, right? So it can't grow inside those cells for some reason. But every now and again on this side, it does cause tumors to develop. And those tumors can then give rise to a large colony of malformed, misbehaving cells, right? And so that's what we found, okay? So in non-target cells, the virus behaves in a peculiar fashion, leading to the production of tumors. We sort of looked at this earlier, so we have a reiteration of this right here. Now, it says 9 properties, but you only see 7 listed. Well, let's have a look at why that is. So we told you earlier that cancer cells or tumor cells are rounded and other cells have their normal morphology. So those morphological changes are very important, right? We also spoke about contact inhibition is removed. So making contact with a neighbor doesn't stop you from undergoing the cell cycle. Related to that, that's why we have 9 properties. We can also say that they have a high saturation density within a test tube or inside the patient. We also spoke about anchorage independence, so they don't need a substrate, the basement membrane or something to be holding onto. They become independent of these growth factors and other types of cytokines and chemokines that are present in the connective tissue. And related to that, they have rapid cell division, even when these chemicals are withdrawn, either in the test tube or within the in vivo environment of a cancer, right? And they have, because of their growth rate, they have an increased need for food, and glucose uptake is increased proportionately. They become immortalized. That means they don't have telomere shortening. They don't come to a standstill or have cell death after a certain number of cell divisions. They can grow indefinitely. And of course, they have the first major step towards becoming carcinogenic cancers by forming tumors, right? And we'll look at that progress one of the biggest things in this class is understanding what leads to cancer, right? Is it a single step or is it multiple small steps? And which steps do you have to go through in order to become cancerous? We'll learn that over the chapters. So the question that we're asking now is, where is the virus DNA, RNA located, right? The evidence from all the research strongly suggests that in those cells that are transformed, the virus nuclear material integrates or gets inserted into the chromosome of the host cell, right? So this integration is very important. So it leads to a number of questions, and the questions are such as, is it the position at which the integration takes place inside your DNA that's going to be responsible for the formation of tumors? Or is it the biochemistry of that DNA or RNA that's going to be responsible, right? So these are big questions, right? RNA, DNA, which one is used for integration? Can both be integrated? And how are they integrated? So those questions are the big ones. And once we discover that, we can then look at the biology. Evidence. I had to take a pause there to see which way to tell you the story. So evidence from SV40, we had the tools in those days to study it. So that evidence was using antibodies and other types of measurement devices, markers. We were able to check for the presence of T antigens, right? So T antigens are proteins that are produced by cells that are infected with SV40. So if you have sera, this is the serum from patients or monkeys that have been infected with SV40. Remember, these experiments were not done on monkeys because they're too expensive, but because SV40 is able to infect a number of different species, we were able to take monkey SV40 and infect mice with it. So these were done on cheaply model organisms called mice. So the SV40 nuclear protein, right, was the result of the presence of the DNA of SV40. Now, when the T antigen was lost from these cells, then there was also a change in the phenotype back to normal. And then when the T antigen was reproduced in these cells, the cells became transformed. So that tells you that something that the SV40 generates, either the protein or the nucleic acid, is responsible directly for transforming these cells in the mammalian species, right? So you can turn it on and off, just like the temperature sensitive experiment that we performed earlier, right? So further evidence in the 1960s and 70s was able to show that whereas SV40 can exist outside of the DNA of the host cell, it was found in the case of these tumorigenic outcomes to be incorporated into the DNA, right? So just part of the DNA. Where's the evidence that this happens? Well, evidence comes in different ways. So the Rous sarcoma virus experiments, we realized early on that the capsid, the virus itself, contains 2 copies of the genome of RSV, as well as some extra proteins that are associated with the RNA. We were able to use some sophisticated experiments to show how the RNA is then able to exist inside a typical host cell. And that's what's going on here, right? And we're using different scientific maneuvers to confirm that the 2 RNA genomes of RSV are associated in this fashion, and they may even be associated with the DNA of the host cell. So that was the initial understanding. It was only later on that we found out that things are not that straightforward. What this is saying is that for some species of tumor viruses, which are based on DNA, they do form circular DNA in the host cells, okay? So those are extrachromosomal. That means these circular DNA are existing by themselves outside of the chromosomes of the host cell. And they can then be passed on to future generations of those cells as they divide by the mechanism that plasmids use in bacteria. So the plasmids are there in high copy number, and when the bacterial cell divides, just by chance, some of the molecules happen to go into one daughter cell and the others into the other. But that's binary fission. How does it work in meiosis and mitosis? Well, it's very similar. If the circular DNA somehow gets attached to the chromosomal DNA, then it'll be efficiently segregated into the two daughter cells. And that's the way that these viruses can continue to remain inside their host cells, right? So we call that autonomously replicated DNA, okay? And you can see here, some of these molecules can be quite large. So everything depends on, so the question is, how do viruses cause tumors? And the answer is, it depends on the type of life cycle and the nature of the host cell, okay? So the most important thing is for us to learn is that there are two types of virus life cycle for normal viruses. So the retroviruses, they integrate into the host cell as part of their normal replication cycle. So the word normal should be highlighted, okay? So they have to do this regardless. And as part of their normal lytic cycle, they enter the cell, they somehow get themselves into the DNA of the host cell. That's interesting. Aren't they made of RNA? So that's something that we have to figure out. Once they enter the chromosome of the host cell, then they need to make copies of that nucleic acid and then somehow get that packaged into the capsids so that the infection can continue into the next cell. So pretty demanding stuff, but we'll see how that happens. This figure was taken from your Introduction to Cell Biology, Bio 2 textbook. And it shows you that these particular retroviruses, they do carry with them in their capsid an enzyme. And that enzyme is very important. It's called reverse transcriptase. And it's an enzyme that can make, from a single-stranded RNA, it can make a hybrid molecule. Not only that, the same enzyme can continue on to remove the RNA by hydrolysis, and then at the same time, make the complementary strand of DNA. So reverse transcriptase is able to do its magic and produce a double-stranded DNA molecule. There's another mechanism available to these proteins, and that is to integrate that newly generated double-stranded DNA into the host chromosome. So this is the amazing part, right? So going back to the previous slide, so the answer to how these retroviruses work, like the Rous sarcoma virus, is that they carry the necessary biochemistry to make DNA, and then the DNA can also be inserted into the host chromosome. That in itself is mind-blowing. How a tiny little capsid not only carries new information in the form of RNA, but also carries proteins that are necessary for these biological functions. Then at a later time, or immediately, depending on the needs of the virus is sensitive to the biochemistry and the biochemical nature of the cell. At a later point, copies of the DNA are made into RNA, just like with producing messenger RNA, or ribosomal RNA to be more precise, right? So the RNA is produced, and then from that RNA, you can take some of those particles of RNA and package them into the heads, and others can be translated using the ribosomes of the host cell to produce the viral proteins, including the reverse transcriptase. And then some beautiful mechanism is able to package into the progeny virus particles the right proportions of all those chemistries. And now the system is ready to infect neighboring cells. The alternative to this mechanism is DNA tumor viruses. So RNA retroviruses, they do what we did on the previous slide. Now we're going to look at the second mechanism, and that's to do with DNA tumor viruses, right? And that would include SV40, right? We spoke about SV40 quite recently. So what happens is that these two DNA tumor viruses, they don't normally integrate into the chromosomes of the host, right? But in non-permissive cells, they may accidentally get integrated into the chromosomes of the host. And you can see here, it's by accident, not by design. So these viruses do not need to insert themselves into the chromosomes of the host as part of their normal life cycle, but it can take place accidentally. And that's the biggest learning thing from this chapter when it comes to the life cycles. So where did these retroviruses come from, right? And what do their nucleic acids look like? So they have the genes for making their protein coats called the capsids. They have genes for making reverse transcriptase, and they have genes for making their envelopes. Remember, the envelope may be different to the capsid. Occasionally, what happens is that the nucleic acid of the virus gets inserted into the neighborhood of a normal gene, or a normal gene is somehow accidentally packaged alongside the chromosome so that you get this hybrid molecule. So these are very rare, but it can happen because the number of virus particles that infect tissue is enormous. So these rare events can happen. So you can see this is the wild type nucleic acid that existed in the lytic cycle of these viruses. And then this is the host chromosomes oncogene, right? C stands for cellular. So if it's a normal gene that doesn't cause cancer, it's going to be called a proto-oncogene. So this is a normal gene that's required for cell division, and you'll see later on the types and classes of genes that these are. Because of this accident of nature, now the proto-oncogene is picked up and packaged into the capsids of the retroviruses, right? And this is how they are now able to transform cells, because they somehow are able to change this gene to become problematic for the cell that it infects. So if I'm a virus and I infect a cell and I'm carrying the V-onc, then I'm going to cause that cell to transform and become tumorigenic, right? In this particular example, we're looking at one possible mechanism by which you can turn a proto-oncogene, which is under normal control, into a superactive gene. And that's what happens when these viruses use this particular mechanism to transform cells. So here we have a virus, a retrovirus, inserting itself into the chromosome of this host cell. And it happens to be using its own promoters to increase the production of messenger RNA from this normal gene. And now that gene has become abnormal in its production of these proteins. An excess of these proteins will lead to a proliferation of that cell through various mechanisms. So you could technically remove the word proto and then call that, it's become an oncogene, right? So proto-oncogenes, when they become mismanaged, their name turns into an oncogene. So the book is full of many examples. So here's one. We'll go back to Sark, right? So avian leukosis virus, ALV, remember that please. It's a virus of birds that causes a blood cancer. And you can see here, the virion in the past, infected this cell, and it happened to integrate just by chance into this location. So are these locations random or are they preferred? We'll look at that in a second. But in this case, the nucleic acid of the virus has inserted itself upstream of the Sark gene. And that's going to now accidentally be packaged into the progeny capsids. So this is going to be a new generation of virus particles that didn't exist before. So this is a type of evolutionary leap. Regardless, when these virions infect other cells, guess what they take with them? They take with them a RNA copy of the Sark gene. And that will then be present in that new cell, the new victim, and that can cause the production of tumors. Do you want more examples? Well, let's give you another one. So in this case, we have ALV, the provirus, going in and sitting down at different locations, right? So this was the example of it changing the level of transcription. So in this case, it makes no difference because these genes are not important for the cell cycle, right? They may be important in some other biochemistry, right? But the c-Myc gene, so here's Myc for the first time, the c-Myc gene is very important as a regulator of the cell cycle. And when the promoters for ALV come and sit down here, they increase the rate of transcription and therefore translation of the c-Myc gene, and that extra protein produced by this cell leads to the proliferation of the cell, right? If you recall from your genetics or your cell biology backgrounds, in eukaryotic cells, we do have a more complicated architecture to our cells, regulation, the gene regulation, than do prokaryotes, right? In the case of prokaryotes, most of the control elements are just upstream of the gene. But in our cells, in eukaryotes, especially higher eukaryotes, we have a more complicated regulation mechanism. We have the proximal promoters, we have the distal promoters, we have enhancers and activators and silencers, right? So sometimes the enhancer may be downstream or even within the gene. So keep that in mind. So if the provirus, which is the integrated version of the nucleic acid of the virus, if it gets into different places, depending on which places it enters, it can also lead to an acceleration of the production of these proteins, which are oncogenic. So keep an eye on that. It doesn't always have to be just upstream of the gene. Sometimes it's within the introns, sometimes it's within the exons, and other times it's on the 5' or 3' end of the gene. So this type of insertion of foreign nucleic acid, DNA, into our chromosomes can lead to the transformation of that cell. So this is a great way to understand many types of leukemia that are due to mutational changes caused by insertional mutagenesis, right? So we have normal cells, and then we have the introduction of a virus, and the virus sits down at a particular location, and that may be random, and there's no change in the behavior of the cells. And then there's more. As the virus spreads from one population of cells into the neighbors, then it inserts into new locations. And as the virus load multiplies within that patient, it could be a bird as well, you find that the sheer number of virus particles is becoming huge. Therefore, the target cells that are infected by these viruses are proportionately becoming more common. And then suddenly you have an insertion at a point that does exactly what we were talking about on this slide here, right? So you get this insertion here ahead of an important gene, and now you have a leukemia. And that is called the malignant phase of leukemia, right? And then you get rapid increase in the number of cells. So you can see it could be a multi-step process leading to the production of a malignant outcome. So you have to go through these other changes. It could happen straight away if you're really, really unlucky. You can get this effect right away if just by chance the provirus enters your chromosome just upstream or close to a proto-oncogene. Other types of viruses are known to be associated with high levels of cancer. So the herpes virus is one of those chronically important viruses when it comes to the etiology of cancer. So some of these are very tumorigenic in animals at a very high rate, as we'll see on the next slide. And when we look inside the tumor cells, we do not find evidence in some cases of the presence of the provirus or even the virus. So the biology may reflect a hit and run hypothesis, where the virus enters the host cell, transforms it, and then the virus is no longer passed on to the progeny cells because it wasn't copied over into them. And therefore, the virus has done its damage and left. And we see the consequences. So here's some viruses that you may be familiar with: Epstein-Barr virus, Burkitt's lymphoma, et cetera, right? So these are pretty severe infections that can lead to tumor development and even cancers. But let's look at a particular example on the next slide. So hepatitis B virus and hepatitis C virus, right? And liver cancer. So here we can see the hepatitis B virus. It is a very important public health issue at this time because around the world, 10% of the population of many countries are carriers. But indeed, let's look at the United States. So in the United States, a significant proportion of the population, normally, unfortunately, in weaker societies, right? Lower income societies, as we should say. There's a problem with drug usage, and that can increase the transmittance of this particular virus. Hepatitis C versus hepatitis B. So in the United States, we have about 3.5 million people infected by both these types. And around the world, you can see the numbers are the opposite. There's more hepatitis B than there's hepatitis C. The average lifetime risk is about 10 to 20% of developing liver cancer from hepatitis C, and only 1 to 5% for hepatitis B. The hepatitis C is a single stranded RNA that doesn't integrate into the host chromosome, whereas hepatitis B does integrate. And the damage on the liver, here there's total death. So if you have hepatitis C, then the level of cirrhosis is approaching 100%. And in these cases, it's slightly below. And the time it takes to develop the cancer can be from 20 to 30 years in the case of C, and between 20 and 70 years in the case of B. But that also depends on your lifestyle, right? People that are more consuming alcoholics, they will have a quicker transformation into the progressive disease. So let's lastly look at therapies that are designed around viruses. So this is where you can use the biology of the viruses to achieve some type of genetic manipulation. The goal here is to take a capsid, remove from the capsid some of the nucleic acids, some of the chromosome of the virus, and replace it with useful genes that you want to introduce into a cell. because viruses are perfect at getting into cells and integrating their nucleic acids, then incorporating your gene into the capsid is going to also introduce your gene into the nucleus. And once that gene is there and it turns itself on, then you're going to produce proteins which could then supplement what the cell was missing. So if this cell was not producing insulin, by putting the insulin gene inside the virus, you can turn this pancreatic cell into an insulin-producing machine, therefore curing that type of diabetes, right? Some of you may know that I got my PhD and postdoc at the Institute of Ophthalmology, which is part of the University of London. And there we did pure research on vision and eyes. And one of my colleagues, Professor Robin Ali, he was in the lab down the corridor, and he was interested in using adenoviruses that were coupled to the replacement of a gene in the back of the eye that causes a retinoblastoma. And he was able to succeed in targeting the retina in the back of the eye with the virus, and having the virus introduced into a few patients, the normal gene, and therefore restoring the protein that was made by these genes and reversing a blind state. And that was touted in those days as a cure for blindness. Did it work? Yes, it did work. It was featured on many news programs in this country when it first came out. But the process has been superseded by other technologies. So even though he's still famous, we have better ways of doing it now because of advances in technology. So this was a bit more risky because it could have introduced, if not carefully monitored, some level of artifact. At UC Merced, there are many strategies taking place right now where we're beginning to uncover new genes that are involved in treating cancer patients directly, as well as using viruses as a vector for delivering those genes into different tissues, right? So if you want to find out more, you should just check the websites of our faculty. There's a lot going on behind the scenes. Until you investigate, you don't know. So now we've reached the end of this particular introduction to this chapter. So now your job is to go back and read the actual pages for yourself and prepare for the upcoming homework. Thank you so much.